Very much to the board for inviting me. I met Don Slinger back in April when I was attending a creation seminar in St. Paul where Jay Siegert was the speaker. And that was the first time that I got to meet Jay. And I'll introduce him further because he's one of the board members for Logos Research Associates, which I am part of now. But I think Don Slinger and Dave Johnson and Ross Olson and the others in the board for having me here tonight. Before we go any farther, though, I'd like to introduce my wife, Elise, who's here supporting me. My wife of 41 years. My sister, Carrie Freilich, who lives in the western suburbs. My son's not here yet. His name is Nathan. He lives here in Roseville, so I appreciate their support. A great friend of mine, Ron Constable, who introduced me to young Earth creationism back in 1971, is here. Ron, hold up your hand. Not only that, he introduced me to the book, The Genesis Flood, by Henry Morris and John Whitcomb, which I read, and then of course, he taught me a lot then from Genesis. We went through the book of Genesis, the first 11 chapters. And of course, having gone to a secular college, I was not a young Earth creationist at that point in time. And all that teaching and that book convinced me. And not only did Ron do that, but he also married Elise and I. So we thank you that, and he taught on the second coming of the Lord. And Don Fidel, another friend, was a housemate when I was at college, is here tonight, and we were housemates back in 71 and 72. So it's good to see familiar friends and family. Today in the mail, I got my Acts and Facts, of which I've been getting every year for about 40-some years. And page 16, genetic entropy points to a young creation. And the first two paragraphs highlight what I'm going to talk about tonight and quote from John Sanford and two publications that I'll introduce you to. So with that, I'll go to my first slide. And that's the book, Genetic Entropy and the Mystery of the Genome, which I put this PowerPoint together from this book. And from Dr. John Sanford, and he originally wrote it in 2005, and then it did an updated version in 2008. And I know Answers in Genesis carries it in their bookstore. And then the other paper is from John Sanford, Jim Pamplin, and C. Roop, Genetic Entropy Recorded in the Bible. And they self-published this through Feed My FMS. It's one of John's foundations. And it's an excellent piece that I also use for this presentation. So these are the board members for Logos Research Associates. I've known John since the late 1970s because of his work in plant breeding. And I actually, after he became a Christian and started publishing things, I was able to review some of his publications before they were published. But I never met him until 2013 when he came to our home. We just live about an hour from here in Amory, Wisconsin. And at that time, he invited me to become a Logos Associate. So he's, like myself, he's a geneticist. He's president of Logos Associates. And as a Cornell University professor, John conducted genetic research for over 30 years. And this research has resulted in more than 100 scientific publications and several dozen patents. John feels his most significant contributions to science was the bialistic process, the book, Genetic Entropy and the Mystery of the Genome, and the development of the Mendel's accountant, an advanced realistic simulation of the mutation selection process. Dr. John Baumgartner is a name that you're probably familiar with because he's been in the creation science area for a long time, a geophysicist. He's vice president of Logos Research Associates. As a research scientist for over 20 years at Los Alamos National Laboratory, he used computer modeling to explore and develop the concept of catastrophic plate tectonics as a mechanism behind the flood, the global white flood. John further applied his expertise in numerous methods in the development of the Mendel's Accountant, the population genetics program for modeling mutation and selection. Jim Pamplin is Secretary-Treasurer of Logos Research Associates, and he serves as the administrator. And then finally, Jay Sagert, who's an engineer, and he's co-founder and principal lecturer for Creation Education Center. And he is the author of Let There Be Light, which is making sense of the creation evolution controversy. He has conducted over 1,000 lectures in 18 different states. In the back of the room, I put one of his brochures out, which is How Do We Know That the Bible is the Inspired Word of God? And he has a number of other pamphlets here. Dinosaurs, where do they fit in the Bible? If God created the universe, who created God? What about the Big Bang? What exactly is carbon-14 dating? Creation in 6 days? The age of the earth? So the one that I have in the back of the room is How Do We Know That the Bible is the Inspired Word of God? Logos Associates, who are we? We are a fellowship of scholars and scientists who faithfully hold to the teachings of Jesus Christ and all of his word, and also faithfully hold to the scientific method and the need for scientific integrity. As ambassadors for Christ, we seek to encourage others to believe in Jesus, to faithfully and deliberately believe what Jesus taught, and to believe God's revealed word, the gospel. We use scholarship, logic, and the scientific method to show that these claims of the Bible are not only credible, but are superior to evolutionary theory to explain the origin of the world we see. And we hope to see all people put their faith in Jesus Christ. Now, I put this slide up there to sort of compare the training that John Sanford went through and myself. There's a lot of similarities in that we both got degrees in plant breeding. John graduated in 1976 from the University of Minnesota. I graduated in 1972. I was in agronomy, John was in horticulture. He then went on to the University of Wisconsin for graduate school, where he got his master's and his PhD in plant breeding and plant genetics. I went to the University of Maryland for my master's, and then on to Iowa State University, where I got my PhD in plant breeding and cytogenetics. John's work was done as a horticultural scientist at Cornell University, where he worked from 1986 to 1998 as an associate professor. He retired from Cornell in 1998. My first employment was with the Ohio State University, where I worked for 6 years. Then I left in 1983 and started working as a plant breeder, soybean breeder, in fact, for Asgrow Seed Company, a private enterprise. I retired in 2010. Let's look at some scripture. In Psalms 139:16, it says, "Your eyes have seen my unformed substance," referring to inside the womb, "and in your book were written the days that were ordained for me." So here the writer conveyed an amazing, accurate concept to illustrate God's awesome wisdom in that God saw me before I was born, and every day of my life was laid out. Only the Creator God could create life, and that life uses the genetic code of life, the DNA, which we'll talk about tonight. In his book, Genetic Entropy and the Mystery of the Genome, John Sanford shows that the primary axiom is false. An axiom is a premise or a starting point of reasoning. The primary axiom is the foundational evolutionary premise that life is merely the result of mutations and selection, natural selection. But John Sanford shows compelling theoretical evidence that the whole genome must in fact degenerate over time, and strongly refutes the Darwinian concept that man is just the result of random and pointless natural processes. This is supported by papers by world-famous population geneticists such as Crow and Lynch, who published in the Proceedings of the National Academy of Science. It is also supported by genetic theory, numerical simulation experiments, and numerous other publications by John Sanford et al. Also, Paul Gibson, who was a classmate of mine at Iowa State University, and here we have John Baumgartner also. So entropy is the gradual decline in disorder. Evolution predicts that over time, because of mutations and natural selection, the best of the mutations will be preserved and the species improved in its overall fitness. But real-life experience and experimental evidence reveals a disturbing downward trend. Mildly harmful mutations are not negatively selected, but accumulate, producing an overall decreased fitness. This is true even though severely harmful mutations are eliminated by decreasing the organism's ability to live and reproduce. And even if there were a few occasional favorable mutations, which is debatable, each species is headed towards decline, which includes the human species. The fact that humanity is genetically degenerating due to mutation accumulation amounts to evolution going backwards. And this is the antithesis of Darwinian thought. Remarkably, such degeneration is very consistent with the Bible. In many places, the Bible indicates that we are a dying people in a dying world and that creation itself is wearing out. Psalms 39:5, behold, you have made my days as handbreadths, and my lifetime as nothing in your sight. Surely every man at his best is a mere breath. In verse 11, with reproofs you chasten a man for iniquity. You consume as a moth what is precious to him. Surely every man is a mere breath. Continuing in Psalms 102, of old you founded the earth, and the heavens are the work of your hands. Even they will perish, but you endure, and all of them will wear out like a garment. Like clothing you will change them, and they will be changed. In Matthew, Jesus says, heaven and earth will pass away, but my words will not pass away. And in Romans 8, this about the whole creation groaning. For we know that the whole creation groans and suffers the pains of childbirth together until now. And the author in Hebrews, you, Lord, in the beginning laid the foundations of the earth, and the heavens are the works of your hands. They will perish, but you remain. And they all will become old like a garment. And like a mantle, you will roll them up like a garment. They will also be changed. But you are the same, and your years will not come to an end. And then in 1 Peter, all flesh is like grass, and all its glory like the flowers of grass. The grass withers, and the flower falls off. But the word of the Lord endures forever. And this was the word that was preached to you. This first part of my presentation deals then with the genetic entropy and the simulation of the Mendel. So here we have some definitions. A mutation is any change in the sequence of DNA. All known mutations cause a loss of information. We inherit mutations from our parents and also develop mutations of our own. Subsequently, we pass a proportion of those on to our children. So as conceivably, in the many generations between Adam and Moses, that a large number of mutations would have been present in any given individual. Genetic bottlenecks or population bottlenecks occur when significant proportions of the population dies or proportions become isolated. Such a bottleneck occurred at the time of Noah's flood, when the human population was reduced to 8 people. Other smaller bottlenecks occurred following the Tower of Babel dispersion, where different people groups were isolated from others. These events would have resulted in a major reduction of genetic variability. These two paragraphs were taken from Dr. George Purdom and Dr. David Menton, who are scientists that work in south of Cincinnati at Answers in Genesis headquarters. The most obvious outward evidence for genetic degeneration is aging, then death and shortened average lifespans. The degeneration of man is explicitly recorded in the words of Jacob, where Jacob said to Pharaoh, "The years of my sojourning are 130. Few and unpleasant have been the years of my life, nor have they attained the years that my fathers lived during the days of their sojourning." So here he's referring to his ancestors who lived much longer lives than he did. And he indeed lived a long life of 130 years compared to our lives today. The extreme longevity of the early patriarchs is very well documented in Genesis, Exodus, Numbers, Deuteronomy, and Joshua. The Bible records the age of death of the first 25 patriarchs in the lineage that goes from Adam to Moses. Here's an answer in Genesis chart that shows the lifespan of man has been declining since the global worldwide flood. The average age of man quickly dropped to less than 100 in just 200 years after the flood. And the flood would have occurred right here before Peleg, and you can see long lives here. Other than Enoch, who was raptured, the average lifespan up here was 912 years. So that's a very long life for those generations before the flood. Methuselah lived the longest at 969 years. And the youngest was Lamech, who died at the age of 777. Then, as you plot out the other ages recorded in the Bible, you can see they drop very quickly after the flood. And then when you get up to David and Solomon, they're about the same ages as the kings of England, from about 1200 to pretty much modern day. So something has definitely caused that lifespan to decrease. Now this is a chart from the paper Genetic Entropy recorded in the Bible, declining lifespans of Noah and his descendants. And what they did here is they plotted out the ages and ran a regression, and they got an R squared of 0.96. R squared is how the data fits a statistical model, and it's explained variation over total variation. When you get that high, it's very, very precise. And it took 20 generations for a new equilibrium of less than 100 years of life to be reached. Moses recorded in Psalms 90, as for the days of our life, they contain 70 years, or if due to strength, 80 years. Yet their pride is but labor and sorrow, for soon it is gone and we fly away. So at the time of Moses, if you lived to be 80, that was considered to be a very long life. The reported decline of lifespans in the biblical accounts were both historically true and accurate. These are real histories about real people whose ages at death were actually faithfully recorded. Given that these early patriarchs apparently lived to know their great-great-great-grandchildren, their old age would have been a matter of great renown, clearly being worthy of being systematically recorded. The shape of the downward slope that we just saw should be immediately recognized by any biologist. It is a biological decay curve. Noah's descendants were undergoing some type of rapid degenerative process. We will show very strong evidence that man is degenerating genetically, and has been for thousands of years, due to continuous accumulating mutations. This makes it very reasonable to conclude that systematic degeneration of man that is documented in the Bible was due to mutational accumulation and the resultant genetic entropy. Now, the Mendel's accountant. This is the comprehensive numerical simulation program that tracks deleterious or harmful mutations as they accumulate in a population, in spite of a strong natural selection. So this is the first chart that was published in this paper, and it's a fitness decline simulation using realistic human mutation rates. And It was conducted over 200 generations of life within a biologically realistic human population, as simulating using that computer program, Mendel's Accountant. As can be seen, mutations accumulate continuously and fitness decline continues. In this time frame, fitness declined over 80%. The result is a classic biological decay curve, very similar to the decay curve based on the biblical longevity data. This simulation employed the known human mutation rate, which is roughly 100 new mutations per person per generation. And they used a population size of 10,000. Each generation, natural selection eliminated the less fit, half of the population's offspring. The blue line represents the population size of 10,000, which is the experiment was held constant from one generation to the next. And the shaded region around the red line is the standard deviation within the population. So biologically realistic numerical simulations show that our current mutation rate, about 100 new mutations per person per generation, human fitness and longevity should have historically followed a decay curve very similar to biblically recorded decline in life expectancies. However, the extreme precipitous decline in lifespans recorded in the Bible just after the flood is actually significantly steeper than our numerical simulations would have predicted. We have reasons to believe that the flood was a high radiation event, and that in the centuries immediately after the flood, mutation rate may have been substantially higher than present. Now here they went ahead and put in that genetic bottleneck of the flood, reducing the population size from 10,000 down to 8, and then ran the simulation. Using Mendel's Accountant, the experiment was conducted to simulate the biblical scenario of mutation accumulation within a biblical time scale of 200 generations, which is roughly equivalent to 6,000 years. The parameter settings used in the experiment were specific to the biblical scenario. Settings included a reproduction of 6 offspring per female, a realistic mutation rate of 100 per person per generation, as well as the genetic bottleneck of going down to 8 people, the size of the family of Noah on the ark. Fitness decline was continuous over 200 generations as mildly harmful mutations continuously accumulated in the population. However, in the 10th generation, there was this extreme genetic bottleneck within the population size. That's represented by the blue line. It went from 10,000 to just 8, followed by a rapid rebound to the original population size. All this did not significantly change the shape of the decay curve. The shaded region represents the standard deviation again, around the line. Conclusions of Part 1. The lifespan data strongly supports the history and veracity of the Bible, especially the book of Genesis. Likewise, biblical data strongly indicates that emerging scientific evidences of genetic degeneration in man are correct, and that genetic entropy is very real. Genetic entropy is the antithesis of evolution and powerfully speaks of the biblical fall. Drastic decline in longevity began very specifically at the time of the flood. This strongly supports the reality of a supernatural, cataclysmic, world-changing flood, not an ordinary or local flood. The declining longevity strongly indicate that the evolution is going the wrong way and that the evolutionary timeline is not viable. All of this points to the desperate need for the redemption of mankind and all creation. Part 2 gets a little more scientific, and we're going to start looking at very minute details. In order to do this, I'm going to use an example of word sentences, chapters, and books, okay? So think of the genome as a library of books. The entire package of information stored in your DNA is called your genome. DNA contains your hereditary information, your eye color, skin color, the shape of your nose, and etc. This library has about 3 billion letters, or nucleotide base pairs. If it were transcribed onto paper, that would fill 200 books with 1,000 pages each according to the Human Genome Project. So letters in a word equivalent to nucleotides, of which we have 3 billion base pairs. Words in sentences would be a grouping of nucleotides. Chapters would equal genes, which are thousands of nucleotides. A book would correspond to our chromosome. In humans, we have 23 pairs of chromosomes. In the crop that I worked on, which was soybeans, there's 40 pairs of chromosomes. In a crop of corn, there's 10 pairs of chromosomes. So different species have different numbers of chromosomes. And the genome then would be the library of all these books. DNA works like a written language, but with very different grammar. Every DNA word is exactly 3 base pairs long, so it doesn't need spaces to tell you when the word ends. It also uses special codes to tell when the end of a sentence happens, and those are called stop codes. There are several different types of point mutations, where you can switch one letter for another, delete a letter, stick in an extra letter, or duplicate letters. The mystery of the genome. Information and complexity, which surpass all human understanding, is programmed in a space smaller than the invisible speck of dust. Each adult human body contains about 100 trillion cells, and every one of those cells has a complete set of life's instructions. The genome is the instruction manual which specifies life. It is dynamic, self-regulating, and multifunctional. It has genes that regulate genes that regulate genes. Every protein encoding gene is embedded within other functional sequences that regulate the expression of the proteins, including promoters, enhancers, introns, leading sequences, trailing sequences, and a sequencing affecting the folding and the DNA architecture. Each cell's nucleus of a human contains 46 chromosomes, or 23 pairs. 22 pairs are autosomal chromosomes, and then the 2 sex chromosomes are the X and the Y. This happens to be a male with the X and Y chromosome. What is DNA? It stands for deoxyribonucleic acid. It's the genetic material in every organism in the 6 kingdoms of living things. And DNA contains all the information that determines what an organism looks like and what it does. And here you see a cartoon of a twisted double helix. In a eukaryote, DNA is found in the nucleus and the mitochondria, and we'll get into that in detail. Okay, here you see a cell with the nucleus, and then the chromosomes within the nucleus, which they pull out one chromosome here, which is wound up in a double helix. And it comes down here, and you see the base pairs are at this end, and I'll explain that in future slides. Each piece of DNA that makes up a chromosome is twisted, wound, and looped around proteins to form a chromosome. Each person has 9 million kilometers of DNA. That's enough to reach to the moon and back 13 times. So if you unwind all this DNA with 100 trillion cells, that's how much DNA is in each person, if you took it end to end. So deoxyribonucleic acid, it's a 3-part unit that makes up the nucleotide of a base, a sugar, and a phosphate. And these nucleotide pairs are also called codons, and they specify whether it's a purine base, it's adenine or guanine, or pyrimidine base, is thymine or cytosine. And A pairs with T and G with C. And each codon represents one of the 20 essential amino acids that are used then to make up proteins. Every 3 letters code for a specific amino acid, such as TGC or ATC, etc. And I have a slide just showing the chart for the amino acids. Cellular proteins are essential for construction, function, maintenance, and reproduction of the entire organism. The DNA molecule is a twisted double helix capable of duplicating itself. So here you see again a cartoon with the different pairs and how they go with the T and the A and the C with the G. And notice that one side of the ladder is going up here and the other side is going down. In the human genome, there are 3 billion DNA base pairs. And this is just a slide showing the 20 essential amino acids and what the DNA codons are that code for those amino acids. Mutations are comprised of nuclear point mutations or mitochondrial mutations, microsatellite mutations, and chromosomal mutations such as deletions, insertions, duplications, inversions, and translocations. So there's different ways that these things occur. The human genome has 3 billion nucleotide base pairs, so there are 3 billion possible point mutations. Every one of those T, A, C, or Gs could become a point mutation. The consensus among human geneticists is that the human race is degenerating. There is consistent accumulation of mutations in the human population, and geneticists widely agree that essentially all these mutations are either neutral or deleterious. Now, genes encode for proteins. The typical whole gene that controls the expression of a typical protein is 50,000 base pair long. So it's a lot of DNA that makes up a single protein. And within the gene, the protein encoding region is typically about 3,000 base pair long out of the 50,000. And the rest of that of the gene is comprised of promoters, enhancers, introns, and leading and trailing sequences. So proteins are large, complex molecules that are crucial to every aspect of the body's structure and function. And there are about 25,000 protein encoding genes in the human genome. An example of some of these proteins would be like collagen, which is important in the structure of your skin and other tissues. Insulin is a hormone that regulates blood sugar. Trypsin is an enzyme involved in digestion. Melanin gives hair and skin its color. And hemoglobin and red blood cells carry oxygen to your cells. And your genotype creates your phenotype by telling your cells which proteins to make, how much, when, and where. The actual rate of all types of mutations may be about 100 per person per generation. There are over 7 billion people on planet Earth today. Each person has added an average of 100 new mutations to the global population. Therefore, those living today have added 0.7 trillion new mutations to the human race. There are 3 billion nucleotide positions in the human genome, so this averages to 233 mutations for every nucleotide position within the genome for all 7 billion people. Because of our large population size, humanity is now flooded by mutations like never before in man's short history on Earth. The consequence of these mutations will become manifested in coming generations of our children, grandchildren, and their children. Yet our children's generation will add even more new mutations, followed by the next and the next. This degenerated process will continue into the future. Genomic degeneration also leads to declining fertility and the inability to have children. Since the genome is degenerating, mankind is not evolving, but is declining. In doing a literature search, Dr. Bergman found in 2004 references to 453,000 mutation hits, but among those there was only 186 that were mentioned as being favorable. All the rest were neutral or deleterious or unfavorable. The 186 mutations were beneficial in the very narrow sense and consistently involves loss of function of the gene. Mutation appears to be overwhelmingly deleterious, and beneficial mutations are too rare for genome building. Due to the enormous preponderance of deleterious mutation, the net information must always be declining. Now, the subject of mutation in the human genome should be approached with sensitivity because people are hurt by mutation. The number of families affected by birth defects is tragically high. 2 to 3% of babies have some type of birth defect. There are thousands of human Mendelian diseases where deleterious genes are passed to the next generation, and I'm going to give some examples of that. Families are impacted by the tragedy of cancer, which is fundamentally the result of mutations within our body cells. Growing evidence indicates that aging itself is due to the accumulation of mutations within our body cells. Now, point mutations are mutations where one letter changes on that DNA sequence. So with this point mutation, we lost the information for one word using our examples of words and sentences, as well as changing the meaning of the sentence. So the overall result is a loss of information. Point mutations can be very devastating. An example is a children's disease called Hutchinson's Gilford Progeria syndrome. It causes children's skin to age, their head to go bald, their bones to develop problems, and their body size to remain very short. Their body parts, including organs, rapidly age, which causes death at an average age of 13 years. An inversion mutation is a segment of DNA in a particular strand that reverses itself. So an inversion mutation would be like taking a sentence and spelling it backwards, and we lose the meaning of the sentence. With inversion mutations, we can lose quite a bit of information. These mutations cause serious problems to the organism. The bleeding disorder, hemophilia A, is caused by an inversion in the factor VIII gene. Insertions mutations is a segment of DNA, whether a single base pair or an extensive length, that is inserted into the DNA strand. It's like placing a word from one sentence into another sentence. This insertion is detrimental in that it makes the sentence meaningless. Insertions generally result in a protein that loses function. Huntington's disease is caused by an insertion mutation. A deletion mutation is a segment of DNA, whether a single base pair or an extensive length, that is deleted from the strand. This would be an obvious loss. We have lost its meaning as well as the words that were in the sentence. Some disorders from deletion mutations are FSHD and spinal muscular atrophy. Frameshift mutations. There are two types, one due to an insertion and one due to a deletion. These mutations can be caused by an insertion or deletion of one or more letters not divisible by 3, which causes an offset in the reading of the letters of the DNA. If a mutation occurs where one or more letters are inserted, then the entire sentence can be off. Not only do we lose the words, but we also lose the meaning behind the words. Frameshift mutations are usually detrimental to the organism by causing the resulting protein to be non-functional. Tay-Sachs disease is caused by a frameshift mutation. Single gene inheritance, also called Mendelian or monogenetic inheritance, is caused by a change of mutations that occur in the DNA sequence of a single gene. There are more than 6,000 known single gene disorders, and they occur in about 1 out of every 200 births. These disorders are known as monogenic disorders. Examples are cystic fibrosis. In the last 2 years, a couple in my church found out that 2 out of their 3 children have cystic fibrosis. And obviously, each of the parents were carriers and they didn't know it. Sickle-Cell Anemia is another, Marfan syndrome, and hemochromatosis. Single gene disorders are inherited in recognizable patterns as dominant, recessive, or X-linked. Polygenic inheritance, or multigenic, are caused by a combination of mutations in multiple genes. For example, different genes that influence breast cancer susceptibility have been found on chromosomes 6, 11, 13, 14, 15, 17, and 22. Examples of chronic diseases that are polygenic are heart disease, high blood pressure, Alzheimer's disease, arthritis, diabetes, cancer, and obesity. And then you have these major abnormalities in the chromosomes. Abnormalities in chromosome number or structure can result in disease. They typically occur to a problem with cell division. Due to the, for example, Down syndrome, that person doesn't have 2 21s, they have 3 21s, so it's called trisomy 21. A common disorder occurs when a person has 3 copies of chromosome 21. Other chromosome abnormalities are Turner syndrome, Klinefelter syndrome, Creutzfeldt syndrome. Diseases may also occur because of chromosome translocations in which portions of 2 chromosomes are exchanged. And then there's the mitochondria inheritance. This type of genetic disorder is caused by mutations in the non-chromosomal DNA of the mitochondria. And the mitochondria is outside of the cell. It's the small, round, or rod-like organelles that are involved in cellular respiration. I said outside the cell, I mean outside of the nucleus, within the cell. Each mitochondria may contain 5 to 10 circular pieces of DNA. Since egg cells, not sperm cells, keep their mitochondria during fertilization, mitochondria DNA is always inherited from the female parent. So based on this, our mitochondria traces back to Eve. Examples of diseases of mitochondria DNA is Leber's, hereditary optic atrophy, an epilepsy, and a dementia. Selection acts on the level of a whole organism and not on the level of the nucleotide. Each nucleotide is intimately associated with all the other nucleotides within a given person, and they are only selected or rejected as a set of the 3 billion nucleotide pairs. Mutation and selection cannot create a single gene, especially when deleterious mutations are factored in. This is overwhelming evidence against what evolutionists believe, that mutations combined with selection have created all biological information. This has been found to be completely false. Mutation and selection cannot stop the loss of genomic information, let alone create the genome. Selection occurs on the level of the whole organism and cannot stop the loss of information due to mutation happening at the molecular level. If mutation selection cannot preserve the information within the genome, it obviously could not have created all that information in the first place. The genome must have been designed and could not have been evolved. Conclusions of part 2. Mutation and selection cannot even begin to explain the mystery of the genome. The genome cannot have arisen spontaneously. It had to have been designed and created. It is also very clear that the genome is not evolving in a forward direction, but is in fact degenerating. Genetic entropy of man is real. We are degenerating. This mystery points to a creator who is worthy of our glory, honor, and praise. In his book, Dr. Sanford says when he has talked about this subject that people sort of get down because it is depressing. Well, yes, man ages are declining and we die from diseases or old age. But you know, in the Bible, it talks about life everlasting in our glorified body. And the Lord Jesus Christ is coming back. He's going to reign on this earth. And life is going to change. So I pulled a couple more verses just to pick us up. Isaiah 65, talking about the millennium when Jesus Christ reigns. No longer will there be in it an infant who lives but a few days, or an old man who does not live out his days. For the youth will die at the age of 100. And the one who does not reach the age of 100 will be thought accursed. So, a young person at age 100, what's an adult then? Okay. Life during the millennium, the earth will have some restoration under the reign of Jesus Christ. We'll have men living long lifespans again. Philippians 3. For our citizenship is in heaven, for which also we eagerly await for a Savior, the Lord Jesus Christ, who will transform the body of our humble state into conformity with the body of his glory, but the exertion of the power that he is even to the subject of all things to himself. So in heaven we'll have glorified bodies that are free of all corruption, and we'll live for eternity with Jesus Christ. Now I put in the references, and they're in very small print, but John Baumgartner, some of these secular people like Crow that published on degeneration of the human's genome, Lynch, and then the very last one is the reference to the paper that was published in 2014, Genetic Entropy Recorded in the Bible by Feed My Sheep Foundation. So thank you very much, and now I can take some questions. The question is, what effect do pharmaceutical drugs have on our genes? I suppose you could also say illegal drugs have on our genes too, right? Both, but pharmaceutical drugs target specific genes that have an impact. Some pharmaceutical drugs target specific genes that have an impact on correcting a problem within our bodies, our physiology. And they're developed that way to be helpful. But because, you know, and I'm sort of outside of my area here, we know there are side effects. Every drug has side effects. So whether those side effects affect the gene or just the cells that are around the point of targeted, or you know, cells way away from the point, you know, we know there are side effects. So there's definitely an impact, and I can't get into more specifics. Maybe you as a physician could. Another question. Well, specifically, I'll try that targeted drugs that are developed for genes, like for some cancers, are not recommended for some people, even though they have issues with that disease, because it could, if they're pregnant women, for example, because it could affect the fetus during growth in the uterus. Another question back there. So his comment regards evolutionists that believe that there are many beneficial, or has to be many beneficial mutations in order for evolution to occur. But when you look at the literature, you know, you see it's just the opposite. It puts them in a real bind, but they continue to believe that. And I, as a plant scientist, and some of my predecessors used mutation breeding to try to find better strains or varieties. Back in the 40s and 50s, when plant breeding was really gaining a lot of science behind it, they used radiation mutations, and it just did not work. Everything that came out of it was deleterious. As a scientist, I use chemical mutations, sort of like the drugs that cause mutations. We used chemicals to try to create point mutations, hoping that you could find a new gene that was resistant to disease or an insect. But again, very rarely was there anything that was worth looking at. Most of that material was dumped after about 20 years of trying, and I don't know of very many people today, after 40, 50 years of trying mutational breeding, have been able to develop products out of that. Another question, and back to lady. Okay. What about the evolutionists that say that bacteria are gaining resistance and they become diseases, or the new strain continues to affect disease on the human population? And I can use a similar example in my own life, and that is weeds overcoming a herbicide. They become herbicide resistant, just like bacteria becomes antibiotic resistant. And those are single gene point mutations usually that occur within those organisms. And it overcomes that particular thing, but as far as the overall organism's ability to have better growth and better life, it's not really beneficial. It's not going to extend further populations. Next question.
Yeah, kind of on that same line with the hybrid corn, for instance. They develop the hybrids and they plant it once, and you get more corn, closer rows of corn, all that. If there's a benefit there, but if you would take that same seed off that cob and plant it again, and keep doing that, would you eventually get to a more negative state of yield?
Yes. The question is regarding hybrid crops like corn, or there's hybrid tomatoes. If you take the seed from that hybrid and plant it and save the seed from that and plant it and on, the yields will go down, or the disease resistance will go away, because these varieties are being inbred, and the more inbred they are, the lower their vigor are. It's hybrid vigor is what creates the high yields to start with, and that's why they take two inbreds and put them together to form a corn hybrid, or two tomato inbreds and combine them in a hybrid. But you can't, if you didn't have the inbreds, you can't create the new hybrid in the future generation. So if you're isolated without the inbreds and you just have that corn hybrid to use, eventually it's not going to be a very productive product.
So a farmer has to, it's a good sales pitch, and the farmer has to go next year and buy a new hybrid.
Yeah.
It's just good for 1 year.
The comment there is that it's a good sales method, because the farmer has to go back to the seed company to buy seed the following year. Question? Yes, the answer is yes. That's why Dr. Sanford said that probably after the flood, and our understanding is before the flood it had not rained until the flood came, and that there was a canopy covering the earth that protected the people that lived on this earth from harmful radiations. When that broke up and the waters came down, there was probably a lot more radiation from the sun that hit Earth, and that's why he says the mutation rate was probably higher at that point in time. And you know, today when we don't guard ourselves, go outside, we get skin cancers because radiation hits in our skin, and we develop skin cancers. So that's why we've learned to dress, cover ourselves to avoid those types of things. So yeah, if you were floating around in outer space without a spacesuit on, you'd probably get, besides dying of cold, freezing to death, you could get radiation poisoning. Way back in the black. Well, I think that's due to modern medicine and you know, protecting ourselves from diseases, eliminating diseases, and two is health and wealth. You know, society is getting better all around the world, and better food and nutrition, and then of course the medicines and the hospitals and clinics that we have, and that not as only in the United States but around the world. So yeah, we have seen an uptick, which is good, because people are living 10, 20 years longer based on the charts that have been kept for many years than they used to. But when you look back, you know, when this nation was founded, a lot of people lived to be 80, 90 years old. And probably there were just less diseases when they came across from Europe and settled in the United States. Question here.
It's an interesting hypothesis by creation scientists that the explanation for Noah's long lifespan is that he didn't have as much genetic degeneration. And it sounds like it actually could be testable. If all life forms exhibit genetic degeneration, then would one not be able to take a look, for example, at the trunks and tree rings that were buried in the catastrophic flood to see if the average age of the plant life was also a lot older than it is today? So have any creation scientists done that experiment or thought about ways to use that experiment as a test of their hypothesis?
So for the video, the question or comment related around the age of Noah at the time of the flood, and how can we go back and look at plant tree trunks and fossilized type things that can be found that occurred at the time of the flood to determine whether life at that point in time was longer in other species besides man. And has anybody done any type of research like that? And I'm not the person to answer that. Has anybody heard or seen any evidence of that type of research being done? All right. Oh, that's true, but he's referring to the time of the flood and later than that, right at the time of the flood. So seasons would have started at that point in time.
Yeah, but also it is true though, what you're saying is true, that you have to postulate that before the flood and during the flood, that there were seasons that could generate tree rings. So if you believe, if the Bible says that's not the case, if the Garden of Eden, for example, did not have seasons, then tree rings wouldn't really exist, I don't think. There would just be, as a botanist, you would probably know what creates a tree ring, but in general, tree rings have an annual ring pattern because the growth rate changes between winter, spring, summer, fall. And it does that cyclically because of the rotation, or so-called rotation of the Earth, and revolution about the sun.
All right, the comments there, again, were wrong. Tree rings and seasons of the question right back with the blue shirt.
I remember a presentation where it was a presentation about a clock in the nucleus of the cell. And this presentation said that if you had perfect medical science and so forth, you eliminate the major diseases, people would live to be 95 years old at the current time. There's a clock in the cell.
The comment was regarding a clock within the side of the cell that determines the age of a man. And your reference was the telomere, which has an impact on that, and whether or not God has adjusted the clock over time. And again, I can't address that subject. The comment was regarding beneficial mutations, and if they do occur, what's the frequency of their occurrence? And if they occur, are they due to a loss of information or a gain of information? And again, the citation, which wasn't that old, where he did a literature review, the beneficial was 6 out of 10,000 mutations. So that means 9,994 were deleterious or harmful. The 6 were due to a loss of information. Now, a loss of information still could allow a bacteria to overcome an antibiotic or a weed to overcome a herbicide. But there's other antibiotics and there's other herbicides that we can use to control the bacteria or the weeds, and that's what man has done. As far as gaining information, that I haven't seen any evidence for that type of mutation. Okay? Question, yellow. 2 parts, but I'll address the last part in regards to GMOs, which stands for genetically modified organisms. And today in our world, corn and soybeans, but not wheat. A lot of people get confused. Wheat has not been modified so that what we buy flour in the stores is not modified. But most of the protein from soybeans and most of the cornmeal would be from genetically modified organisms. What impact does that have on the human being that's consuming that? When you Google the subject, you come up with all kinds of answers, anti and pro, okay? I'm a scientist who worked in the area and bred with genetic modified genes, and they were highly tested in the organisms years before they were released into the public. The first organisms was tomatoes and soybeans back in the mid-90s, so they've been around for 20 years. And all the tests showed that they had no impact on the food that was being consumed, not only by humans, but by the animals that consumed the food, then we consume the animals. So the genes were always taken from another plant and not from a mammal or from a human. In those cases or bacteria, they were taken from bacteria, but not from humans or animals. So from my point of view, from being a scientist working in the area, they're not harmful. and I have no problem eating and consuming genetic modified meal from protein or corn or soybeans. Yes, sir. Well, that is false because they've been buying our soybean meal for the last 20 years. Barges and barges have gone over, shiploads have gone over to Europe. What the Europeans have not allowed their farmers to use it for in their own fields. There has been testing done in Europe. I personally conducted tests in Europe for seeing if the crop performs as well as the non-GMO crops. But as far as the using it on their own farms, they don't want that. They want other parts of the world like South America and North America to grow the crops and send it to them. They don't grow enough food in Europe. They have to use food from outside of their countries in order to feed their populations. Yes, definitely there's that kind of work going on. There's work trying to develop transhumanists and all that type. There's a lot of futurist scientists out there that could go over into the deep side as far as I'm concerned. But as far as gene therapy, I think there's a place for gene therapy for some of these diseases that have no way of resolving an issue on. And if it could introduce something that could affect the gene so we could produce the needed counteractive thing to the disease, that would be beneficial. Again, anybody from the medical side want to address that? And he just commented that it is being done today, gene therapy to counteract negative diseases on people. Yes, ma'am.
Isn't it rather naive to think that GMOs are not going to be harmful to the human race when they've only been around since the mid-90s?
As far as human GMOs, but viruses have moved DNA around species during the 6,000 years of life. So DNA is still being transferred, it just hasn't been done by humans. Yes?
How about chemicals just sprayed on the plants themselves? Is there any effect from that?
Like the herbicides and the fungicides? Well, the negative effects is what you're talking about. The question is, spraying herbicides or fungicides have any negative effects on plants? I'm just trying to think of some negative effects. I mean, they were created for the beneficial effect to kill the weeds or to protect from a fungus or a bacterial or a viral disease.
Or to affect humans, right?
Well, yeah, well, if there's still remnants of the, especially insecticides, on the plants and we consume that, yes, that's definitely negative to humans because it wasn't designed for humans to eat it. So that's oftentimes farmers are given on their instructions, you put this on so many days before harvest, so it's not there by the time harvest comes. So if farmers don't always abide by those rules.
Wash your apples.
Yeah, wash your apples. Yes, sir.
I was told by a friend who's a farmer, maybe 3-4 years ago, that they were inserting the genes of a pig into corn, and he thought this was wonderful to prevent, I'm guessing, pests from attacking it. Are you familiar with that?
No, I'm not. From a pig into corn, no. Most of these genes are coming out of bacteria. I'm not familiar with that particular case. If it was, I would share it with you. Well, transposons, when I went through graduate school, is also called jumping genes, where it can move a gene from one part of the genome into another part of the genome and can affect the phenotype then, resulting what it looks like. And in corn, it could make it look like Indian corn, where you have all the different colors in the kernels. That's one example. And your question then related to, can that have an effect on diseases? Right, so transposons can move into different parts of the genome. They can turn on a gene or turn off a gene, and depending whether that has a positive or a negative effect on a disease. So yeah, those things do occur, and they're natural. It's not man-made. Most of the major world crops are being looked at for genetically modified organisms like rice, wheat, even though I said wheat isn't there yet. They are being looked at. in some of your major vegetable crops. So again, you have to look at it from a company's perspective. It can't be a minor crop because they have to regain all their research dollars that they put into it. And it costs like $1 billion to introduce a new crop with a specific genetically modified trait in it. So it has to be a major crop. So if it's a minor crop, it's probably not going to happen. Your question?
Do you think they'll ever find a cure for diabetes?
Will we ever find a cure for diabetes?
Type 1 or type 2?
Type 1 or type 2?
I would think so. I think there's so much work going on that type 2 would be a trait that could be cured. I hope so. I have diabetes. Yes.
I have a question about Francis Collins, the person who led the team that mapped the human genome.
Yes,
I understand he's a theistic evolutionist
that's correct.
and claims to be a believer. Do you know if he has confronted this problem of entropy? Do you have any of his writings or speeches where he might deal with this?
All right, so the question revolves around Dr. Collins, who was the director of the Human Genome Project. He is a theistic evolutionist and supposedly a Christian, as to whether or not he has been personally confronted on the subject of genetic entropy. And that'd be a good question for me to ask John Sanford, because I don't know the answer. But I'm going to ask John that very question, because that may change his mind on theistic evolution.
I heard him speak, and this was about 10 years ago. It wasn't long after the genome was mapped. And he said that our genome is 95% similar to that of the chimpanzee, and we most definitely are related.
Well, that's creationists and scientists have now disproved the 95% similarity, and it's down somewhere around that 70% number, isn't it, that I recall. So we're not as similar to the chimpanzee as they originally thought, because when they did the comparables originally, they did a subset of the DNA. They didn't do it of all the genome. Okay, and when you do all the genome, it's closer to 70%. So that's a lot of dissimilarity. There's thousands of genes that are not similar in the species of monkeys or apes and humans. A question over here.
What are the major arguments against this genetic entropy hypothesis that you've heard?
No, I mean, most geneticists will agree to the subject that there's a degeneration occurring. Even in plants, I see it. If you grow a variety, year after year after year after year, it's accumulating mutations. In the University of Minnesota, Dr. Rasmussen did a paper on small grains showing that mutations occur over time in a variety. And you look at different species, like dogs. I mean, some dogs have some leg problems or hip problems that other families of dogs don't have. And you know, the cats, wild cats. You know, why did a lot of species die out over time? And we've lost a lot of species. Was it due to genetic entropy or hunting? I think it was due to some of both of those things. So, you know, I think there's lots of evidence for genetic entropy occurring. And again, when you look at the literature, geneticists agree with the subject. They just come to a different conclusion. Yes, Nathan, my son.
So now that the human genome is mapped, which is a fairly recent occurrence, we have technology like supercomputing where you can do a lot of these, you know, calculations of these billions of base pairs. How much do we understand at this point about what we can turn on and off in the, you know, the genome to affect different diseases and kind of engineer our way out?
So the Human Genome Project was done. It's been probably 15 years. It cost billions and billions of dollars. The science and the technology today is that you could do that human genome for probably less than $5,000 And thus they're doing more and more people, like Dr. Watson's DNA has been sequenced. And that the genome then gives us a starting point to search for the genes. Without the genome being sequenced, we had no way to really search for the genes. And with that, scientists, doctors are finding genes that affect, what you're talking about, turning on, turning off, different physiological processes. But it's a very slow process still to find the genes and then find out what the gene does. And slowly it's coming along. So every once in a while you'll hear in the news, such and such a person found a gene that impacts this disease of humans. And once they find the gene, then they're going to start targeting a cure for it. It's slow.
One more question.
All right, you can have it.
Do you think that genetic entropy is responsible for things other than some of the diseases you've talked about, things like the increase in allergies, increase in depression, increase in anxiety, those kinds of things?
Okay, so the question was, besides the diseases I talked about, does genetic entropy have an impact on other things like allergies or depressions or autism or all that stuff? And I would think that it definitely does, but there's also environmental effects that we as a civilization have changed the environment around us so much that we've probably caused more things to impact our genome. So you have the genetic by environment interaction, which creates some of these side effects. So the answer is yes, and that's the last question.
Well, thank you very much.